Welcome to the video demo for the Exploring Cancer and Nitrate in Drinking Water app. This app is designed to be an investigative experience, allowing anyone to explore the relationship for themselves. The key thing I focused on was really providing context and walking the user through what's happening. Now, I won't read everything aloud on the page, but to start us off, let me at least go ahead and read the introduction. Nitrate is a contaminant which can get into your drinking water supply because of fertilizers and other waste. We can use data from Wisconsin to help figure out an important question. What is the relationship between nitrate in drinking water and cancer? The methodology section explains how we can statistically prove some sort of relationship. In order to see how nitrate and cancer are related, we will run linear regression on statewide data from Wisconsin. Results of the regression could tell us how much of the cancer rate can be explained by nitrate concentration in drinking water. So we have our independent variable, which is the nitrate concentration. As I mentioned, it's from water well samples in parts per million. And then we have our dependent variable, the variable that we're trying to explain, which is cancer rate from census tracts. Unit is percentage of population. Now, the end user, doesn't need to know the full analysis that's happening. In fact, the only parts they interact with are these three parts highlighted in green: the interpolation, the residuals, and the results. But for posterity, I'll briefly explain the analysis. So we start with cancer rates in census tracts, as mentioned, and nitrate concentration in water well samples. The key here is the regression. So in order to do the regression, we need to get the data in the same format, which is what this interpolation and zonal statistics is for. So the interpolation estimates the nitrate concentration for a whole area, basically using those water well samples and estimating basically between them what those values might be. And then from there, we can use zonal statistics to summarize that interpolated surface back into census tract zones. And now that we have both cancer rate and nitrate concentration in census tracts, finally we can run our regression. Now, everything I just explained is in this little text here and also in this drop-down explanation box, so users can read all about it if they need to. So the second section we have here is the analysis section. We have as I mentioned, 3 points of interaction. We have interpolation parameters, which is how we actually run the analysis. And then we have our 2 outputs, which is the map of regression residuals down here in the lower left, and the regression results on the right. Both of them are empty currently because we haven't actually run the analysis yet. As you can see, there are messages in each saying, run the analysis to see your regression results. So we'll go back up and do that. The most important part of the interpolation here is the power. So if we kind of zoom in here on the power and smoothing graphics, it's kind of a very simple explanation, but just to give the user an idea, power, it tells the interpolation tool basically how far of a reach should each of those wells have? How much influence should they have over their surroundings? So if you have a really high power and really high smoothing, you're going to see a surface that looks more like the surface on the right here, totally kind of filled in, and those wells having an effect over the whole area. So let's zoom back out, and I'll go ahead and run. I'll just use 1 and 1 for the power and smoothing. They can both be anything over 0. And I'll hit run analysis. We can tell it's running by the little spinner and the fact that it says running analysis over here in the log. And when it's done, just like that, it'll say done with the inputs that we used. I do want to go ahead and run it with the defaults though. So I'll just change power to 2 and rerun the analysis. Again, it says running analysis. We see a bunch of data down here now, and it just updated because our second analysis just finished. And let's actually go ahead and look now at our results. So on the left here, if I could draw your attention, are the census tracts that I kept mentioning. These are the real ones. And for each of these census tracts, we have a residual amount, and that's all the colors you see here is it's a map of the residuals. And a residual is just a fancy way of saying the error. All this map shows us is where the regression model over and under predicted, right? So I'm currently hovering over one of the tracts here in the upper right, and the information for it is shown in the upper right of the map. So the actual cancer rate for this census tract was 0.08. The estimated cancer rate, our estimated cancer rate was 0.078, which means the residual is negative 0.002. That is a really close prediction, and we did really well for this census tract. But if I just move one over, suddenly the residual is a little bit higher. It's a little bit further from zero. We didn't do quite as well with this census tract, but that's okay because it's still relatively close, if you ask me. Now, really, the point of looking at a map like this, a map of residuals, is to look for clustering. So if we're trying to make like the perfect model, a map of residuals like this, we wouldn't want to see any clustering, because basically if our model is tuned right, the residuals would be random, right? If we see clustering, it means that we're missing something. I'm okay with missing something for this analysis because we're only using one explanatory variable to try to predict cancer, right? We're only using nitrate concentration in drinking water, and we know there's a bunch of other things that can cause cancer. So I'm actually content with the fact that it looks relatively random, given some exceptions, such as these urban areas, which are kind of consistently underpredicted. Like you see down here in the lower right, this is Milwaukee, and then kind of closer to the lower center, that's Madison, pretty consistently under estimated. But overall, it looks decently random, decently good. So let's draw our attention over here to the regression results. Now, the regression summary here, these results, they really tell us two important things. Number one, what the R-squared value is. That's this in the upper right. And number two, if we can trust the R-squared value. That's basically the job of everything else here. The R-squared value in this analysis, it was 0.32, suggesting that nitrate concentration in drinking water could explain 32% of the cancer rate per census tract as long as the R-squared value is trustworthy. How do we know now if the R-squared value is trustworthy? Well, let's look at the other factors in the regression summary. Because the p-value here, probability of the F statistic, because the p-value is close to 0, and the F statistic is really large, we can reject the null hypothesis. Basically, these two prove that there is a linear relationship between the independent and dependent variables. Now let's go and look at this mean row here. Mean here refers to the mean or average nitrate concentration per census tract. So if we look at the t-value, the 25 here, and also look at the p-value, which is 0, we can reject another null hypothesis. This proves that the nitrate concentration is significant in the prediction of cancer rate. So what all that means is that we can trust our R-squared, and what we can say is that given the statistical significance of the regression model, the results show that nitrate concentration in well water can explain 32% of the cancer rate in Wisconsin. And that is an impactful statement, and I hope that users will be able to explore this analysis to their content. If they want to run it again and try to get different R-squared different values, they can change it up and rerun to their heart's content. And my explanations that I mentioned are also available in these drop downs for when I'm not there talking to people. That is all. Thank you.